Good morning, good afternoon, good evening. Hello, my name is Garrett Morris. I wanted to present my IS Symposium poster and give a brief overview of the topics that I have discussed and examined in my IS project. So this is Wriggling Through Toxins, an investigation of the protective role native microbes may play in the C. elegans detoxification response to neonicotinoid compounds. So to briefly touch on the background, I wanted to discuss the prevalence of neonicotinoid pesticides. So neonicotinoid compounds are insecticides that specifically target insect pests, usually sucking, biting, and root feeding insects. And they act on a specific receptor called nicotinic acetylcholine receptors and cause death by overstimulation of this receptor. And they are specific to these insect NACHRs and do not target any other species. These neonicotinoid compounds are the most highest widely used insecticide today. And it's important to study the effects and sublethal effects that these may have on organisms that aren't their target species or non-target organisms. There's been a slew of research that has been on the non-targeted effects on many other species, including fish, other pollinators like bees, and in some cases, humans in acute cases. I wanted to discuss and examine the role the microbiome can play in protecting C. elegans, our model organism, against these toxins. So I wanted to use C. elegans because there is a robust catalog of microbial organisms which for exists a large characterized set of microbial organisms that we can easily manipulate within C. elegans, making it an ideal candidate for this type of examination and analysis experiment. I also wanted to present the methods that I've used. So to maintain C. elegans, I used N2 C. elegans on NGM plates and kept them alive using an OP50 E. coli food culture and maintained them throughout my study. I performed synchronization experiments to get my C. elegans to the same life stage by using a hypochlorite solution containing bleach to kill all the live worms and leave only the eggs remaining. And then I did two very specific assays that allowed me to quantify the physiological effects and therefore the neurological effects and effects on fertility, those being movement assay to quantify the neurological effects and an egg-laying assay to quantify the fertility effects. And as you can see here, I have a diagram, a brief overview of part of the process that I used for my movement assay or locomotion assay. I started by plating my C. elegans from a synchronization containing a bacterial food source, which is one of their native microbiome strains, the toxin, which was one of two neonicotinoid toxins, imidacloprid or clothianidin, and then my eggs. I let those grow for a period of time, and then I transferred them using a worm pick onto a plate that was enabling me to quantify their locomotion in distance traveled towards a food source. So some of the important results and conclusions that I found were that C. elegans, when exposed to neonicotinoid toxins, they experience defects when they do not have presence of that toxin there. And in addition, I found that native microbiome strains provide better resistive effects to these toxins than do that of our control strain, which is E. coli OP50. As noted in figure 3, we can see chronic exposure to neonicotinoid toxins results in a higher presence of locomotion for a specific strain, a native strain called MyB71, which is higher than the remaining strains, suggesting that this strain may play a role in the detoxification response. In addition, below that figure, we see when exposed to clothianidin, all 3 of our native chosen microbiome strains present with a higher level of egg laying than our control strain OP50. And some of the implications or discussion points of this study are, how does this work? What is the mechanism driving behind it? I'm not going to get into the whole detail. You can stop by my poster and ask me questions about it now, but a brief overview of the discussion points that I want to discuss are different microbes native to C. elegans may be playing a role in different effects on their detoxification response. In either, they may be doing this of one of many ways, and they can use metabolism of these toxins, reducing them to a less harmful state for the worm, or they could be affecting C. elegans' innate defense responses through some form of gene expression manipulation. And secondly, a little more specific, a target gene that I wanted to examine and I think may be involved is the SKN1 gene, which is interesting to me because it has a human ortholog called NRF2, which can suggest some trends in the human detoxification response. But there will be more studies needed to confirm and test this hypothesis. I'm very excited to present this at the IA Symposium. Thank you so much for listening, and I hope you have a great day.